Okay, welcome everybody to the 5th session of the Global Ocean Oxygen Network webinar series, or the GO2NE webinar series. We are very happy that you're joining us today. My name is Lisa Levin. I'm a professor at Scripps Institution of Oceanography at UC San Diego in the US, and I will be your moderator today. And let me start with some housekeeping. You, as the attendees, have the option to type your questions in the question box, and please use the chat to share additional information or let us know if you have any logistical problems. If any of you are interested in speaking in one of the upcoming GO2NE webinars on a topic related to oxygen, please feel free to submit a short abstract, and the link for this will be shown in the chat box. So I'd like to begin. We have 3 wonderful speakers today. We're going to start with Lillian Mccormick, who will be presenting for 20 minutes, followed by about a 10-minute question and answer session. And afterwards, Karin Limburg and Liz Lojudis will be talking again, will be speaking together for about 20 minutes, again with a 10-minute question and answer session after. So let me start with Lillian Mccormick. Lily is a postdoctoral scholar at UC San Diego. She's working with me at Scripps Institution of Oceanography and with Dr. Nicholas Esch in the Department of Psychology. Lily obtained her Bachelor's of Science in Marine Science from Eckerd College in St. Petersburg, Florida in the US, and her PhD from Scripps Institution of Oceanography at UCSD. Along the way, Lily's worked on many things, including the effects of oil and dispersant on zooplankton behavior and physiology after the Deepwater Horizon oil spill. But for much of her time in recent years, she's been looking at the effects of oxygen loss on sensory physiology and behavior in marine organisms. And I'd like to mention that if you like what you hear from Lily and have a hiring opportunities, Lily will be available and is looking for her next position in the coming months. Okay, Lily's talk is titled "The Blinding Truth. Vision is highly sensitive to oxygen loss in the ocean." So please, Lily.
Okay. Thank you so much, Lisa, for the introduction, and thanks to everybody for coming to hear this talk and for inviting me to speak today. So as Lisa mentioned, today I'd like to talk to you about why vision is so sensitive to any change in oxygen in the ocean. Okay, so why do animals actually need vision
Lily,
in the ocean?
I'm sorry,
yeah.
we can't see your slides right now. We just see your white work before. I know.
Okay, let me try one more time.
Now it's perfect. Thank you so much.
Okay, you can see that okay?
Yeah,
okay, sorry about that, everybody.
Sorry. One more. Now it's presenter mode.
Lily, can you go to presentation mode?
Swap display.
How's that?
Yes.
Okay,
no, swap display. No. Again, swap display.
No?
It doesn't work right now. Yeah.
How about this?
Yes. Perfect.
Okay, thanks everybody for your patience. Okay, so let's bring us back. Why do I want to study vision in the ocean? So marine organisms use vision for a wide variety of reasons, but largely this is to find and capture prey items, to detect and escape from predators, to find conspecifics, so animals of the same species for mating or for communication. And also light is a primary cue for vertical distribution for many marine organisms in the ocean. However, vision has extremely high oxygen requirements, and this is primarily due to the fact that aerobic metabolism fuels ion transport in photoreceptor cells, which are also just called visual cells in the eye. And so this has been really well described in terrestrial vertebrates, but not so much in marine organisms. And if we were to hypothesize what animals might be most affected by this, we could say that taxa with fast vision might be the most susceptible to visual impairment from oxygen changes. And when I say fast vision, I really mean animals with highly sophisticated eyes that have extremely high spatial and temporal resolution. And temporal resolution is essentially just how fast the eye is able to process something. And so in the marine environment, this really means that we're looking at active arthropods, cephalopods like cuttlefish and squid and octopus, and then also marine vertebrates or fish. And shown here in this table, marine invertebrates and vertebrates have different eye types as well as different mechanisms for processing their visual information. However, that's not super important for today. I'm just going to focus on marine invertebrates for this talk. Although currently some of my postdoctoral research is looking at this, whether oxygen affects vision in fish. Okay, so I probably don't need to explain in depth to this audience why ocean deoxygenation is such an important aspect of climate change. However, globally, we've seen about a 2% decline in the oxygen content in the last 50 years. So this figure is showing oxygen loss in these warm colors, and in these cool colors are indicating areas where oxygen might actually increase in the future. Or has a history of increasing. Some other studies have kind of broken this down to, despite the fact that we see about a 2% global decline in oxygen, regionally, some of these oxygen losses can be much more severe. And so this figure is showing that in different regions of the world, oxygen loss can actually, in certain regions, can actually range from 20% all the way up to 50% in different areas. And this is incredibly important for the organisms that live there because they might have special vulnerability to this oxygen loss that's occurring at a much faster rate. And you know, this figure also gets into the drivers of deoxygenation. And while I'm not going to have time to discuss that today, it is an important consideration for why these losses are happening. So today I'm going to focus a little bit on my research, which is based off of Southern California in the United States. We call this area the Southern California Bight. And this is one of the regions that's experiencing pretty high oxygen loss over a very short amount of time. So my research focuses on the question of, does low oxygen affect vision in marine invertebrates? To do this, I'm focusing on marine larvae in the Southern California Bight, which is the area I just described. And this area is really important because both oxygen and light conditions vary on diel, seasonal, and interannual scales. The marine larvae that live here vertically migrate from about the surface to 50 meters depth, but definitely are not usually found at greater depths than about 100 meters. And importantly, just like their adult counterparts, these marine larvae rely on vision for prey capture, predator avoidance, and also light is their vertical migration cue. So today I'm going to focus on 4 different individual species: the California market squid, the two-spot octopus, the tuna crab, and then the graceful rock crab. So my research really focuses on the impacts of 2 stressors, oxygen and light, on the visual physiology of animals, the metabolic physiology, the visual behavior, and then I subsequently tried to see how this might affect the visual ecology of these organisms. The first question that I wanted to ask was, how does low oxygen affect visual physiology? So how do I actually test vision in marine organisms? I use a method called electrophysiology, which is essentially measuring the summed electrical response of visual cells or photoreceptors in the eye. And this is called an electroretinogram, or ERG. And this electrical signal essentially tells me how well the visual system is functioning. So I'd like to remind everybody, these marine larvae are exceptionally tiny. They range in size from about 2 millimeters to 4 millimeters. So all of my experiments are conducted underneath a microscope, at least for this section. So here in this image, you can see my setup. This is the microscoped objective coming from the top. Here you have this oxygen probe that sticks into the microscope stage. And the microscope stage it holds about 4 milliliters of water, and I'm constantly perfusing the stage with new seawater. And the important thing is that I can control the chemistry of that seawater. On the left and the right of this image, you can see 2 micromanipulators, and one is used to tether the individual larva while I'm doing the experiments, and then the other one is used to measure the electrical response in the eye. And you can see an image of that in this bottom photo right here, with the electrode coming in from the left. So what does this actually look like? One of the nice things about this setup is that I can control the light stimulus. And so if I give a repeated light stimuli of increasing intensity, shown here in these red bars, I get a corresponding electrical response from the eye. And nicely for me, the electrical response is greater if I have a larger stimulus, at least here in these marine invertebrates. So essentially, I can use these to characterize the visual response of these organisms. So for the general setup of the experiment, I put the animal on stage, make sure I have a happy, healthy larva. All of these studies are conducted in vivo. And then I make sure I have a stable visual response. And then I start decreasing the oxygen that the animal is exposed to until I reach a certain low oxygen level. I keep it there for some time and then start re-oxygenating the solution. And today I'm going to talk about what happens on the decline when I'm lowering oxygen. Okay. So in order to actually measure this visual response and also compare across species, I needed to come up with 3 visual metrics to basically define and quantify the results that I was seeing. So V90 is the partial pressure of oxygen at which there's 90% visual function remaining. V50 is where there is 50% visual function remaining. And then V10 is the pressure at which there's 10% visual function remaining. And essentially what this allowed me to do is normalize the response to the greatest response at normal oxygen conditions for each species. And so then I could compare, even though the visual responses looked a little bit different. So what did we actually see? So for each of these plots, this is oxygen partial pressure on the y-axis, and I've shown the mean points of each of the 3 visual metrics, V90, V50, and V10 for all 4 species. What we saw is that cephalopods and the brachyurian crab, shown here in the cool colors and then also the orange, show extreme sensitivity when I reduce the oxygen level in the chamber. And the oxygen levels at which I see sensitivity are at conditions that we think are greater than hypoxia, which is typically defined at about 5 kilopascals or about 60 micromoles per kilogram. And really importantly, we saw some really cool species-specific sensitivity within the visual system. So if you can see the market squid and the graceful rock crab, as soon as I started decreasing the oxygen in the chamber, the visual response also started decreasing. The 2-spot octopus was a little less sensitive. The visual response stayed the same until I reached a certain oxygen level, and then the visual response started tanking and decreasing. However, the tuna crab we found was relatively insensitive, so much so that we were never able to define a V10 oxygen level because the visual response just didn't get that low. For all of these 3 sensitive species, by the time I reached the lowest oxygen condition, they really did not have any visual function. They were almost essentially blind. And so this shows us that visual physiology is extremely sensitive to this oxygen loss. With that information, the next thing I wanted to see was whether this physiological impairment from exposure to low oxygen would then be manifested in their visual behavior. So how does that affect the visual behavior of the organisms? And I did that by studying something called photobehavior, which is essentially directed movement due to a change in light in the environment. So the way I set up these experiments was put squid larvae in this vertical chamber. And it might be a little bit hard to see with the shared screen. However, each of these tiny little black dots is a squid larva. And they're inside the chamber and they're swimming around. And so what I can do is essentially provide this light stimulus for these organisms and then see how they change their behavior in relation to the light stimulus. So for each of the experiments, I held the animals in about 2 minutes of darkness just to look at their swimming behavior and see where they distributed themselves in the chamber. I then exposed the animals to 9-10 second light stimuli of different irradiance levels. And then again, followed by another 2-minute control of dark behavior, swimming behavior in the dark. And what we really wanted to look at was how the larvae changed their vertical position in the chamber. So this is all in millimeters, from positive 80 is representing the top of the chamber where the light stimulus is, 0 is the center of the chamber, and then minus 80 or negative 80 is the bottom of the chamber. And what we were really interested in doing is comparing the vertical position of the larvae in the 30 seconds before the termination of the light stimulus. So that's 20 seconds in the dark and a 10-second light stimulus, compared to the 30 seconds after the termination of the light stimulus. And that's shown here in these yellow and purple bars. And I'll explain that a little bit more later. And then we completed these experiments at 4 different oxygen levels for each species. And I measured both the octopus and the squid larvae in this. For brevity, I'm going to talk about the squid larvae, or the results from the squid larvae today. Okay, graphically, what does this actually look like? Here are some results from these photobehavior experiments. So here we have the mean vertical position of the squid paralarvae in the chamber as a function of the experiment time in seconds. And so this is the entire experiment with the 2-minute dark period, and then each of these vertical orange bars is a 10-second light stimulus with the irradiance displayed up on the top of the graph. And so essentially what this black line is showing you is the mean vertical position of all of the squid in all of the control chambers, or all of the control trials that I ran. So this is under normal oxygen conditions, just to get a read for what the visual behavior of these organisms is. And so what we found was that there's this really robust characteristic sinking behavior after the termination of the light stimulus. And we call this the off sinking response. And so this is represented by these dips in the vertical position shown with these arrows here. The nice thing about this is that this occurs at every single light stimulus we gave them, but it's more apparent at higher light stimuli than lower light stimuli. So at high light, this behavior is really large, and then it's smaller at smaller light levels, which was nice and allowed us to quantify the difference between the behavior 30 seconds before the termination of the light stimulus, shown here in this yellow bar, compared to the 30 seconds after the termination of the light stimulus, shown here in purple. Okay, bear with me here. These are the actual results from all of these photobehavior experiments. And so let's focus on this top left plot. Here I'm showing the vertical position of the paralarvae, all of the larvae in the chamber for the squid, now as a function of irradiance. And so you see high light levels on the right and low light levels on the left. And again, the yellow boxes are the behavior 30 seconds before the termination of the light stimulus, and the purple bars are the behavior 30 seconds after the termination of the light stimulus. So what you can see is clearly what we saw in the other figure, where you have this characteristic sinking behavior after the termination of the light stimulus. And so you can see these larvae are sinking, and the sinking behavior is magnified at higher light intensities. And here also the red asterisks indicate significance between those two 30-second periods. So when we start decreasing the oxygen to V50 levels, which, these correspond to the visual physiology oxygen levels, what we see is not that much different. We still see this really robust sinking behavior, even though it might be slightly changed. However, when we start decreasing the oxygen even more, you can see this behavior starts to disintegrate. You see a lot fewer significant differences between the behavior in these two 30-second periods. And then by the time we're exposing the animals to the lowest oxygen conditions, this behavior has completely been destroyed. We don't see any photobehavior at the lowest oxygen levels. And really cool is that the oxygen threshold for this photobehavior occurs at partial pressures of oxygen that equate with about 50% visual function. So at oxygen levels, that would cause about 50% visual function in these organisms. And the cool thing is, I'm just showing the squid behavior here, but the interesting thing is that we also saw the exact same oxygen threshold for both squid and octopus. And that's the 50% oxygen level relative to each species. So the oxygen level is different, but it's about when the animal would experience 50% visual function. Great. So what can I actually do with all these laboratory determined vulnerability thresholds? So here in this table, I have all 4 species that I tested and each of the kind of physiological metrics. So the V90, V50, and V10 from the physiology experiments, oxygen level at which photobehavior is affected for squid and octopus. And then I'm not going to talk about it today, but we also measured the P-crit levels of these organisms in both squid and octopus. And so essentially what I wanted to do is look at the interdependence of light and oxygen and how that might affect organisms. So I came up with a concept called the luminoxyscape, which are combinations of light and oxygen in the environment. When I combine these physiological metrics that I determined with environmental data, I can then determine what's called a critical luminoxyscape, which are combinations of light and oxygen in the environment that enable full visual function. And we really wanted to answer the question of whether oxygen or light could potentially constrain larval visual habitat. So another wonderful thing off of Southern California, we have this great program called CalCoffee, the California Cooperative Oceanic Fisheries Investigations. And they conduct surveys, oceanographic surveys off of Southern California, and they've been doing so for 7 decades, which provides us with this really rich data set of how oxygen and light conditions have changed in the past and also in the current conditions. Okay, so what this allowed me to do was combining these visual metrics with this environmental data, I was able to come up with what the maximum depth of this critical luminoxyscape, where the animals would have visual function. And so for each of these figures, bear with me again, I know this is a lot on a screen, but let's focus again on the top left figure. Here I'm showing depth descending as a function of distance from shore. So on the left is closer to shore, on the right is farther away from shore. These color scales indicate the oxygen levels. So warmer colors are higher oxygen levels, cooler colors are lower oxygen levels. And this orange line is showing the irradiance threshold of the visual physiology. So essentially the lowest light level at which these animals can see. The purple line is showing the maximum depth of occurrence of the V50 oxygen level. So essentially the oxygen at which each respective animal would have 50% visual function. Okay, so what do we actually see? We see that vision becomes incredibly oxygen limited near shore for all 4 species. So you can see these habitats seem to be a little bit, these visual habitats would seem to be a little bit visually, vertically compressed near shore. And most importantly, we found that the sensitive species are more likely to be oxygen limited for vision rather than light limited for vision. And let me explain what I mean here. So let's look at the market
Lily,
squid.
I'm going to need you to wrap it up fairly quickly. I'm
Okay,
being told
no
we're running out of time. So.
Okay, sorry about the technical difficulties at the beginning. So essentially, the market squid and the graceful rock crab are going to be oxygen limited for vision and not light limited, but the less sensitive species are more likely to be light limited than oxygen limited. And I didn't talk about the P-crit today, but the larvae would only experience P-crit at depths of about 300 meters for the squid and greater than 500 meters for the octopus, indicating that these are vision is probably more likely oxygen limited than metabolic physiology. So I'm just going to quickly put up my conclusions here. We did see that vision was highly sensitive to oxygen loss and at oxygen levels that are present in larval habitat right now. So again, sorry for the technical difficulties and thank you very much. I'd be happy to take any questions.
Thank you, Lily. And I'm really sorry to cut you off a little bit.
Had some technical difficulties at the beginning.
We've started to get some questions rolling in. Nancy Rabelais asks, how do these results interact with change in the depth distribution of oxygen minimum zones and visual performance in eutrophic or eutrophied waters?
That's a great question. Thank you for that question. So the trick is that we don't really know what the actual vertical distribution of these larvae is. And so that's one of the challenges with this is that right now it's very predictive. But you can imagine if you have an area with high eutrophication, that depth oxygen level or the depth at which you're going to have low oxygen is going to be much shallower than you might have otherwise. And so in Southern California, we have this upwelling, which brings really low oxygen to much shallower depths than you would see, let's say, on the east coast of the United States. And so, but certain events can cause even lower oxygen. So for example, we had a red tide, which caused an incredible oxygen bloom where we had supersaturation in the water column. And then we had an amazing oxygen drawdown where we saw that oxygen went to levels extremely low, even in very, very shallow environments. So that's a really good point. We don't quite know how this is going to affect the actual vertical distributions of animals, but we can expect that in areas where you're going to have high eutrophication or consistent red tides, that's going to pretty vertically compress the animals to shallower areas. Hopefully that answered most of that question.
Okay, we have quite a lot of questions, so we'll see how many
Great.
we can fit in the next few minutes. The next is by Benjamin Walther. Do you think the visual sensitivity would change depending on prior dietary or nutritional histories or other metabolic stressors?
I do. Yeah, that's a great question. So there is some evidence that photobehavior changes in starved larvae. And so essentially just that photobehavior can kind of be changed, either reversed or just not as strong when you starve the larvae. So kind of when they don't have enough energy, theoretically, for their visual systems to function. I don't think that paper made that connection, but they did show an effect where starved larvae, it does affect the photobehavior. So that's a really good question. So yes, I think it would. Yeah.
We have a talk from a question from Karin Wishner, who says, great talk. Do animals in nature avoid the low oxygen or become incapacitated?
Ooh, that's a really good question. So I don't totally know the answer for these marine larvae. There has been a lot of evidence on both accounts. So some marine organisms are really good at avoiding these low oxygen areas, while other marine organisms can actually utilize these low oxygen areas to kind of avoid predation or something like that. I don't know the answer for these larvae. My hunch is that they would probably try to avoid these areas, but again, I don't have a lot of data to back that up. So great question. That's something we need to look at in the future.
Andreas Ashley says, thanks very much. Do you see signs of adaptation if you look at species living in naturally different oxygen conditions?
That's a really good question. I would assume there would be. The closest thing I saw to adaptation is that scientists know that tuna crabs are incredibly tolerant to low oxygen, and that's partially because of the areas that are in their normal habitat. So typically they'll live in very low oxygen habitats. What was really cool is that I saw that the larvae of tuna crabs were really, really low tolerant to low oxygen as well. We previously had only shown that the adults are very tolerant to low oxygen. But I did see that there wasn't as much of a change in visual function with the tuna crab larvae when I exposed it to very low oxygen conditions. So that might indicate some form of adaptation that's present in the animal. So that's a great question. I think there will be adaptation.
We have a question from Naya Latinen. Sorry if I got the name pronunciation wrong. She asks you, what prompted you to focus on vision when it came to your research regarding hypoxia?
Also another very good question. So I kind of grew up in a vision lab. My undergrad and some of my research during my undergrad, as well as afterward, my undergrad mentor studied vision in marine invertebrates. So I was very interested in vision already. However, when I was research teching before my PhD, I started working on experiments looking at how hypoxia changed the growth and behavior of fish, just killifish in the east coast of the United States. And I kind of started putting two and two together and trying to look at any research that had been conducted about marine organisms and oxygen and vision, and kind of, I knew vision was very sensitive to oxygen from other research, but we hadn't seen if that was true in marine organisms. So I just started kind of combining all of the research that I was being exposed to and developing this hypothesis. But good question.
Okay, I'm going to apologize to the 6 or 7 other people who've submitted questions that we're not going to get to. Clearly, there's been a lot of interest, so thanks so much, Lily,
yep.
for speaking with us.
My email is on the screen. If you'd like to email me questions, I'd be happy to answer them.
Great.
Thank you, Lisa.
I'm not sure if you can stop sharing, but I'd
Yes,
like to go ahead and introduce our next presentation, which is a dual act by Karin Limburg and Liz Lojudis on a realm they call environmental communication. So let me introduce both of them. Karin Limburg is a State University of New York Distinguished Professor in the Department of Environmental and Forest Biology at the SUNY College of Environmental Science and Forestry. Karin received her bachelor's degree from Vassar College, a master's of science at the University of Florida, and her PhD from Cornell University. She's also a visiting professor at the Swedish University of Agricultural Science and an adjunct at Syracuse University and Texas A&M Corpus Christi. So she's a lot of places. Her work embodies economics, ecology, and physics, and more. And she will be presenting today on one component of her project Breathless, which explores multiple dimensions of hypoxia. And she will be presenting with Liz Lojudis, who is an environmental educator and communications specialist from New York State's Hudson Valley. And Liz is currently principal of Residence Communications and Consulting, LLC. She earned her bachelor's degree from the State University of New York at Albany and a master's of science from Green Mountain College in Vermont. Liz is interested in multidisciplinary collaborations that produce effective methods of communicating scientific concepts in ways that inspire audiences to take action. Liz is a musician with a keen interest in using sound to enhance environmental understanding. And this is reflected in the title of their talk, The Sound of Hypoxia, Turning Cod Otolith Data into Sound and Song with Data Sonification. So Karin and Liz, please.
Thank you. I might need a little help here with actually getting my screen to show. Is it showing, folks?
Yes,
we can. Okay, very good. So you're seeing
you
this?
make it full screen?
It is now. Is it not for you?
Not yet.
Nope.
How about
Presenter
now?
mode.
Let's see. Well, I'm in sharing, and
You go to your PowerPoint and just make it presenter mode.
let's see. Well, I'm in presentation mode right now.
Okay.
Can you
Yeah,
see
it's
that?
fine like that. It's fine like
Oh,
yeah.
Is it okay or is it not okay?
We still can see your sidebar with the slides.
Okay. Well, let's just try it by going the. Well, let's see. I never use this. I just always use the. I just use F5. I just do it that way. But let's. How about now? Success or not?
It doesn't
No,
change.
but we can see it, so it's
Yes,
probably
it's okay.
going to be okay.
All right. Let me just try one more time. Let me try launching it again. And let's see. I think is the slideshow mode one of these little things here? That's it. Is that better or not?
It's not changing, but just go ahead.
All right, we'll just go ahead then. Okay.
Yeah.
In that case, all right. So thank you very much, and Lily, very, very nice talk. And I hope that we can do a good follow through here. So our presentation that Liz and I are doing is a part of a project that's sponsored by the National Science Foundation of the U.S. on, as Lisa said, the multiple dimensions of hypoxia. And one of the things that we're very interested in is really finding out how people, how we can actually communicate this to people. Aside from all of the actual science that we're doing, we actually want to know also if we can make it understood for many publics. And we have a website, which I'm showing the web URL below here. And I did not put my email on anywhere, but our nickname for the project is Project Breathless. So data sonification, it's actually kind of too bad that you that we have not in presentation mode because this is actually animated, but we'll just go ahead anyway with it. So you.
I'm sorry, Karin. I will, because we don't see how you change your slides.
Yeah, your
So
slide is advancing.
Are you seeing slide 2 or not?
No. So I'm taking back your presenter rights,
Okay.
and then we'll give it back to you. Okay?
All right. That sounds good.
Okay.
All right. So shall we try it again?
Yes, I'm doing it now. Sorry.
Okay. All right. We'll see if it works this time. So I will try it in screen share. Can you see that?
Perfect.
Is it good? Okay,
Yes.
That's great. All right, so what we're doing for this aspect of our project is something that's called data sonification. And Liz will talk a little bit about it. We probably won't have time to talk in great detail about it, but hopefully we'll have time and questions. But it's actually used quite a lot now. It's used for some data interpretation applications. It's used to detect some patterns. That's one of my interests, and also communicating to non-scientific audiences. And so as part of our project, we have been interested in, oops, sorry, I think I just, are you still seeing my screen?
Yep.
Yeah, okay. As part of our project, we're sonifying some data from codfish from the Baltic Sea, and we're testing out some contrasting stories of different degrees of hypoxia exposure. And just FYI, I've been studying cod in the Baltic Sea for quite some time with respect to hypoxia impacts. But what we're doing here is really trying to understand about messaging. And messaging is all about trying to get out relevant points that you want to be able to have many audiences understand. And in our case, we want to know if we can create effective messages about hypoxia in this way. And so we're kind of at the beginning of this part of the project. Liz, did you want to say something here?
Yes, I just wanted to add that sonification it's a non-speech way to display data in a sonic way, and it's part of a larger field called auditory display. And so Karin and I are really novices in this field. We recognize that there are many artists and scientists who are much more accomplished in creating music inspired by data. But we're, as educators, we're really intrigued with the possibility of using song and sound to translate information and stimulate action. So that's really our motivation. But we're on a learning journey with this work.
Yep, thanks, Liz. So the object of our study is literally the object is something called an otolith, and otoliths are actually part of the hearing and balance system in fishes. And this is a photo of an otolith of a cod, and this is kind of how they're positioned in the brain case. They sit underneath the brain, connected with the semicircular canals, and they perform function in hearing and balance. But for me and others, I think I've begun to think of them as kind of the ultimate logbook for each individual fish, because we have both chronological or chronologically marked information in chemistry of these objects. So otoliths are calcium carbonate concretions that are or structures that are precipitated on these scaffolds of complex proteins. And they've become extremely useful in fisheries science for their chronometric properties. But what we can do here, for example, with the cod otolith is we can make a cross section through it like this. And when you do that and polish it down to the core, we can see actually an entire lifetime recorded here. So we actually have a record that goes from the start of life at the core all the way through as it accretes through its entire life. And with the contrasting translucent and opaque zones, this has become very standard fare for age determination. So there are probably something like a couple million fish a year that are aged in this way. But there's also chemical information that's locked up inside. And I don't have time to go through everything. We're going to just try to make that part very simple today. But just to say that it is the chemical information that we are using in our sonification. The star today of this is the Baltic Sea Cod. And Baltic cod are in actually a very terrible state for the most part today. They've been endemic in the Baltic for a long, long time. Right now the populations are really threatened. And hypoxia is one of the threats, certainly. So Dan Conley showed maps like this last week or last month, and what we have for looking at the Baltic proper are maps that are now created every year, but they go back to 1960, and they show us the extent of hypoxia in gray and anoxia in black. And the period of 1993, the early 90s, were the lowest period in that record of hypoxia and anoxia. And unfortunately, now we're seeing more and more of this kind of, like the panel below. And fish that are in good condition would be nice and fat like the one on the left. And on the right, unfortunately, we're seeing increases in fishes in poor condition. Maybe not always that skinny, but pretty skinny. Dan, and we also have a means to try to push the record back. And Dan, I think, talked about this last month as well, having baselines that we can push back into the early part of the 20th century to see that, in fact, hypoxia was much less frequent at that time. But we can even, with fish, if we're lucky, push it back even farther. And I have been lucky, and I've been working with an osteoarchaeologist with some Stone Age otoliths, which we've been also probing. And so these are, this photo shows 3 of those with a modern otolith, and also a chunk of manganese. And manganese is our Hypoxia proxy that we use in otoliths. So I've begun to think that nowadays I can ask the fish to tell us the answers to these questions. For each individual fish, where did I go? What did I see? And maybe even, was I happy? So what we decided to do was to test otoliths of 5 different fish, 2 of them being what I call happy cod, so the cod that were living in normoxia. 1 of them is from the late Neolithic, and 1 is from the early 20th century. Then a fish from that low hypoxia period in the 90s, but I'm not going to have time to play today. And then an unhappy fish, shall we say in quotes, that experienced severe hypoxia for much of its life. And then another one of those that was caught in 2019, but had the great fortune to be transported to an aquarium at the Baltic Sea Science Center in Stockholm. And it lived in good water quality, ate all the food it wanted, it lived a good life. So Liz, I think you're going to take this one.
Yes. So just very quickly, I wanted to talk about the steps we went through to sonify these data series. They are very long. So when I first sonified the data series, we had a song that was about 30 minutes long, which was a length that even we didn't want to listen to. So we figured audiences might not be that thrilled to listen to 30 minutes of our data. So we had to decide how to shorten the length of each data series without losing detail and nuances of the data. And we are particularly interested in maintaining the seasonal patterns of the fish's life that you see, as you see depicted on these graphs. So all the way on the left, you see the full data set for phosphorus in the otolith, and we ended up averaging every 5 rows, which is the middle graph, and it shortened it, but kept the structure, and that came out to about 6 minutes long. And so then we tried again, averaging every 3 rows, which you see on the top right, and that was still a little lengthy. And so then we went for another 5-row average, which you see on the right on the bottom, which we felt that approach made the data short enough to be listenable. They're about 1 to 2 minutes long without sacrificing too much information.
Okay, then you're taking this too, is that right?
Yep, so we just wanted to share the chemistry that we sonified and a little bit on why and how we did that. The magnesium, the ratio of magnesium to calcium is a function of metabolism, and we regard it as our metronome of the fish's life. And when the fish is in normoxic waters, that chemical indicator is played as marimba. And when it's in more hypoxic water, we have it playing as piano. The ratio of phosphorus to calcium is related to growth, and it's played as a trumpet. We also have strontium, which relates to salinity, and it's not featured in every song, but that's also played as a trumpet. And barium, the ratio of barium to calcium, which indicates terrestrial influence, is played as an organ. And as Karin mentioned, our hypoxia proxy is the ratio of manganese to magnesium, which is played on violin and piano and is only present, obviously, in fish that have been exposed to hypoxic waters. And this data is processed very heavily with digital effects, and in which we're trying to help people relate to the way that a fish might, the sluggish, disoriented way that a fish might feel when swimming through low oxygen conditions. And we also worked with tempo and the mode, major versus minor, the time signature, and other factors to accentuate the sense of well-being that a fish might feel in adequate oxygen, as opposed to the dis-ease of swimming through hypoxic water.
Thank you, Liz.
Sure.
So we're gonna, I'm not sure if we'll actually be able to play all 4 if we're really held to the time. Maybe we'll play the first and the 3rd and 4th.
Yeah, we're about 6 minutes.
Yeah, okay. Yeah, so so we'll see. But this is a slide that's full of otoliths that I analyzed from the Neolithic period. And the one that we're going to hear today is this fish right here from about 4,000 or close to 4,500 years ago is when it lived, and it was hunted and eaten, and yet its chemistry survives. And so the data sets that we're going to do from this fish are 3. We picked out strontium, which is in the purple here, and it's related to salinity. As we explained before, so it's positively correlated with that. And the Baltic is actually kind of a big estuary. It was actually a little bit more saline in the Neolithic period than it is today. Barium, as we mentioned, is kind of an indicator of terrestrial influence, particularly early in life. And this high barium and low strontium indicate, on the left here, indicate residents of the young cod in what we call a nursery area, kind of a nearshore nursery area. And then as you can let's see if my mouse comes back. If you follow through the first, this is kind of like the first year of life, and then somewhere in the 2nd year of life, it makes this directed dive into deeper, more saline waters, and then apparently it comes back. And so on, and the magnesium just kind of clops along, and so that's why we decided to use that as a metronome. And so that's what you're going to hear. So I'm going to now have to figure out once again how to switch screens so that I can play this song for you. So this is now going to be the song of this fish. And I'll try to follow along also with my mouse. So here goes.
You're muted, Karin. You're muted.
Oh, am I muted? Can you hear me?
I can hear you now.
Okay. So I think I'm back on the slide again. So it wasn't that glorious the way this trumpet blared when this fish made it out into the ocean. I thought that was really cool myself. And there is actually some harmony in there, which is very interesting. It's nothing that we really did. This one here, I will talk through it, but it's one I don't think we have time to play. I certainly will arrange for private showings if people want. But this is a fish that also lived the good life. And what was interesting about this one is that, again, this idea of the metabolic metronome, very, very regular. This fish lived in Normoxia and had a really kind of regular metabolic, or what we think is a metabolic a rhythm throughout its life, 9 years old. And if we were to play the song, which takes a couple of minutes, we would be hearing kind of that steady rhythm. The phosphorus, on the other hand, is very it's very regular, but in a very different way. And playing the two together actually makes some very interesting some very interesting harmony as it proceeds. But unfortunately, I don't think we have time for this one. So we'll skip that one and go to the next one. So cod, this cod was caught in 2014, and this fish was exposed to severe hypoxia. During the last maybe 10, 15 years, the hypoxia and other conditions in the Baltic have become so extreme that cod don't lay down clear annual marks anymore. And I have placed marks on where the chemistry tells us winters occur, but you can't tell that visually on this. And so my student Yvette Heimbrand is actually studying this problem for her PhD, but this particular fish I analyzed myself. And what we can see here is a way, just a very crude way of taking a look at how the Hypoxia Proxy would play out. We can just sort of superimpose it on a flattened out image of the otolith and then just sort of mark off the calendar years of where this occurs. And so we can see that this fish every summer experienced hypoxia, and particularly extreme probably in its later years. Without going into great detail, we've defined thresholds that are age-based for hypoxia occurrence. And so I've colored those here in red. For those of you who are colorblind, it's black probably in contrast with the blue, that's normoxic. And then I also color-coded the magnesium, which you're going to be hearing as well. And what's interesting about the magnesium is it sort of cycles along in that clock-like way until about here, and then it kind of picks up and then goes into what I can only call a tailspin. And it's decoupled from the seasonality of anything else, except for the hypoxia is the only thing that really persists with that. Liz, were you going to take this one?
Yeah, just very quickly, we used a really user-friendly, beginner-friendly app called GO2NE to sonify the data. And there are some limitations to that app, and one being that it correlates pitch to numerical amounts. So greater amounts of manganese, which indicate greater exposure to hypoxia, result in higher pitches, which we felt was good for conveying the sense of anxiety that may result from having difficulty breathing. But it doesn't do such a good job at conveying the sluggishness that an organism would suffer from ongoing hypoxia exposure. So we ended up inverting the hypoxia proxy so that we could play the hypoxic periods in a lower pitch and hopefully have people relate more. And we did a number of other manipulations with the data, the tempo, the time signature, the mode, etc., to convey what a fish might feel in hypoxic water.
Right, and remember also that this is what we're trying to do is convey a kind of a feeling to the public too. And although we won't have time probably to play even the last one, I will play this one, if I can get the screen to work on it. And then I'll talk just quickly about the last one as well. So this is the song of the Hypoxia Cod. And what you're going to hear is a change in tempo, which you might pick up particularly around here in the magnesium. You'll hear it kind of quicken, and as we're trying to convey the sense of anxiety, and then you'll hear it slow down towards the end. So we'll play this now. That is the song of the Hypoxia Cod. And we only have about a minute left, so I'm not going to be able to do the last. But I do want to just quickly just scoot through it and say that we are going to be doing a collaboration, I think, with the Baltic Sea Science Center at Skansen, which is a very, very famous park in Sweden, in Stockholm. And just to show you that the cod that are there are very nice and fat today. But they were actually very small when they entered. So I know I don't have time to go through the rest of this and play the video for you, but one last thing is to just say that if you can see my screen, I've got the Hypoxia Proxy chemistry data here, and the fish that were put into Skansen have far and away the highest, some of the highest hypoxia records that I've ever seen. So I think this is evidence that things are worsening. So with that, I'll just say thank you. And I don't know if we have time for one question.
Well, we're supposed to end now, so thank you very much. I have to say, I'm sure for like for me, as everybody, this is a first for all of us who care about oxygen to be able to listen to it. And you've had so many comments in the questions about how fantastic this was. But let's try to squeeze in one or two really fast questions. We have a question from Michaelia Wibawo. She says, hello, I have a question. What is the consideration to decide when to sonify otolith chemistry, and why phosphorus to calcium and strontium to calcium played as the same, or played as the same instrument? Will it not make some confusion?
We didn't play them, I think, in the same song, with the same instrument. We're limited in terms of the instruments that we can use. Why, and thank you for your question, and why we chose these is because this species in this hypoxic area is undergoing such severe stress from hypoxia that it really represents strong extremes. It's a very well studied species. We're very fortunate to have materials, specimens that represent these strong contrasts. And if people want to hear some more of it, I guess we'll have to invite you to come to Sweden this summer to listen at Skansen. Anna Björn is the director there, and I'm sure she'll welcome you.
Thank you. Nancy Rabelais says, incredible stuff. To reach and engage with a large portion of the population, what about hip hop?
We need to change the tempo then, right?
That might be our next endeavor. We'll see.
We in our project, we had said that we would try to compose music. This is music from the fish, but we could put it into other composition. Liz is a musician.
I'm afraid we're out of time. Many people have said brilliant, fantastic, congratulations. I think there's a lot of excitement here. Some people want to know where they can hear these. And I want to know, when is your first album coming out?
Oh, right. We have to figure that out.
So I'd like to thank all of you for attending, to thank all our speakers today, and to encourage everybody who's still online to please join us at the next GO2NE webinar, which will be on April 28th. So thank you, Lily, Liz, and Karin. Thank you so much.
Thank you for hosting us.
Thank you for having us.
Thank you, everybody, for joining. See you next time.
Thank you. Bye.